As I mentioned, this is the 10th webinar, and again, a common request is people to have two things, is to have certificate for their CME, and we will be able to issue certificate for all people who have registered. If you attended without registration, we don't really know your details or your email address. In this case, just please get in touch with me if you have already attended. The other thing is the feedback, and traditionally, also most webinars don't have feedback, but we felt that it would be really useful. And therefore, you will have a Google form nowadays where you can actually get emailed to you to fill the feedback. These webinars have been taking place every other weekend on a Saturday at 11 o'clock London time. And I'd really like to thank the Arab Society of Fetal Medicine and Surgery for offering us to use their platform, which is this Zoom platform free, which allows us really to have a very large number of people joining for limitless time. And actually that was free. So I'm very grateful to Dr. Shadi for actually helping us. Okay, so today the program is usually the webinar lasts for about an hour and a half, and we've got a fantastic speaker who I have great respect and admiration, is Dr. Tessa Humphrey, who is a consultant clinical genetics. She works both at St. George's Hospital and also at King's College. And Tessa is the one that whenever we don't know what to do, we don't know what this condition could be, we always call her, and she's always available, and she always ready to to be available and see the patient and give second opinion, even for actually cases from abroad. So people who are on the webinar that even from abroad, Tessa has certainly in the past helped me with some referrals or second opinion from abroad. So this is going to talk to us about genetics and multiple pregnancy, and I'm really grateful to you, Tessa, and I really look forward to hearing your talk. Thank you.
Okay, can everybody hear me? If everyone can't, can you send a note on the chat? I'm not quite sure what I can do about it, but Asma was cutting in and out a bit for me. Was she? I don't know if she was for everyone else, but so I hope that you will be able to actually hear me. So talking about genetics and multiple pregnancy, I wasn't, the slide isn't going on. Let's try the new slide, yeah. So I just wasn't sure, yeah. So I wasn't quite sure what we were, Asma was hoping for, but of course, dizygotic and monozygotic twins are incredibly different. The incidence of dizygotic twins is twins with 2/3 dizygotic twins. And the interesting things about the dizygotic twins is they're very variable in different parts of the world. So the incidence in Africa is 10 to 40 per thousand, with in Nigeria it goes up to 50 per thousand. And the white population, Asian population, is only of about 3 in 1,000. We know the incidence increases with age, and there is a genetic element to it, with a positive maternal family history of twinning increases your risk of conceiving twins. Whereas in monozygotic twins, the actual incidence is pretty constant over the world of about 4 in 1,000. And it is rarely, although occasionally there is a positive family history, and those are quite interesting, those families. And I'll talk a little bit more about them as well. And there is an increase with both in IVF. Now, it's obviously very easy to understand why there's an increase in dizygotic twinning, but I think the monozygotic twinning is incredibly instructional when we look at the increased risk of that in IVF. So what am I going to talk about here? Because I wasn't actually quite sure what Asma wants. We know that there are certain genetic diseases that couples know that they're going to have, and there are difficulties in testing in multiple pregnancy, but that's really your problem, not my problem. There are obviously the problems of specifically knowing which twin you've actually tested, And of course, selective termination, if that is where you go, is obviously got problems, and the gestation. So I'm not going to talk more about that because obviously that is what you know about. I'm going to talk about fetal abnormalities, obviously in multiple pregnancy. And also what is even more interesting to me is the discordance in fetal abnormalities in monozygotic twins. So, sorry, I have to keep pushing on in there. So in dizygotic twins, the rates of abnormality is relatively similar to brothers and sisters, but there are, of course, exceptions. There's an increased risk of preeclampsia, increased risk of IUGR and prematurity. So there are increased risk of handicap in dizygotic twins. There is some evidence of increased overcrowding, and so you might get an increased risk of talipes. But fundamentally, the underlying risk of abnormalities is similar to single pregnancies if you exclude things like preeclampsia and IUGR. So what about my monozygotic twins? Well, one has to remember 1/3 of monozygotic twins are dichorionic diamniotic. And of course, you miss that in fetal medicine. You do not know if this is a monozygotic or dizygotic twin, if they're dichorionic diamniotic. And if they don't look alike and they have discordant fetal abnormalities, you presume not correctly that they are dizygotic twins. But 2/3 of monozygotic twins are monochorionic diamniotic. And of course, these are the ones that interest you guys the most because of twin-twin transfusion and the complications associated with that. More twinning of monochorionic, monoamniotic and conjoined twins are very rare and account for probably no more than 1% of monozygotic twins. So we don't always know what is the zygosity of the twins. Now, there aren't many animal models of monozygotic twinning, and only the 9-banded armadillo, which incidentally is similar to humans. Same mutation. Always fascinated with animal studies, how they choose a certain animal for it. But the 9-banded armadillo always has identical quadruplets. And there must be a way of finding the gene that actually causes that. But it is the only model. So monozygotic twinning is incredibly rare in most animal species. And so humans are unusual with this. So there is a genetic familial monozygotic twinning, but it is very rare. But the very interesting issue about familial monozygotic twinning is there is no increased risk in fetal abnormalities in this particular group. So if you have familial monozygotic twinning, you don't need to, they're not going to get twin-twin transfusion syndrome. They're not going to get the complications. They're going to be dichorionic, diamniotic twins. So that's very interesting, suggesting a very different mechanism to what we see. Their inheritance is both maternal and paternal, but it can miss generations. So it's not absolutely certain what the cause of that is at the present time. Now, when does twinning occur? Now, this diagram, which is fundamentally quoted in every single paper about timing of twinning, was created in 1961 by this chap called Corner. And he said in that original paper, and the trouble is most people do not go back to the original papers, is once we are able, or if we are ever able to undertake IVF or be able to fertilize embryos in the test tube, this can be challenged. Extraordinarily, it has not been challenged in spite of over 30 years of IVF, and this has to be wrong. And the reason it has to be wrong is that you see dichorionic, diamniotic twins in IVF pregnancies, and they are put back at the blastocyst stage. So there has to be, this is wrong, and that's incredibly important when you are looking at the abnormalities that you see in twins. So implantation takes place here, and then this thing called X inactivation, which I'm going to come back to later on. So let us look about what actually happens. And this is something that you will all be very familiar with, that at ovulation, the egg is actually released. But what you're probably not quite so familiar with, it is only at that stage that the first meiosis 1 is actually completed. So at ovulation, meiosis 1 is completed, and the first polar body is excluded. Now, although the first polar body is, and this secondary oocyte is haploid, it has got 2 sets of chromatids, so it is effectively diploid. And then at fertilization, so you do not complete the second part of the meiosis until the egg is actually fertilized. So if you don't actually go on to fertilize an egg, meiosis 2 does not get completed, and the second polar body is then extruded. And then the two pronuclei come together. Now, so polar is incredibly important, because actually the second polar body, it's haploid, it's in very close proximity. The sperm has actually is around it, it's lurking on the outside. And so there is a very strong possibility, and it has been proven that we can get polar body fertilization. But polar body fertilization will not lead to monozygotic twins, because they are different. The actual chromosomes are different. So they would actually be identified as dizygotic twins if you did genetic studies. So what else can you get from polar body fertilization? And there are various interesting problems, and there's a digynic hydatidiform mole, so you get fertilization, the second polar body doesn't get excluded, and you get a triploid pregnancy. You can then get, and we've got an ongoing patient with this at St. George's at the moment, which made me interested in this, is that actually there is a twin pregnant placenta. So we have, on genetic studies, it looked as though this was a triploid baby, but in fact the baby looks completely normal. And in amniocentesis, the baby has normal chromosomes, but there is also this diandrogenic hydatidiform mole, and it looks as though it was probably the polar body excluded all chromosomes. So there are extraordinary things. And then there's a chimera where there seems to be, so instead of getting 1 for 2, here you've got 2 for 1. So the second gets absorbed into the first. So we're not quite sure how much of the above is caused by fertilization of the polar body, but it does, that we know the second polar body remains alive for some time after fertilization, and it contains little cytoplasm and few mitochondria. So it's going to be much more likely to have abnormalities than is the main oocyte. So what else is absolutely critical when we're thinking about twinning? The maternal genome is directing the first few cell divisions. Well, this is actually essential for all forms of knowledge about genetic abnormalities. And we know that in familial hydatidiform mole that you can get autosomal recessive mutations within the mother's genome in NLRP7 and a few other genes, and they get recurrent hydatidiform moles. So the first few cell divisions are not under fetal control. And the fetal genome doesn't take over until about day 3 to day 5. So we know that the first few cell divisions, where the cells are all exactly the same, they are totally equal, but soon those cells have to commit to some form of gene expression. And that is said to be something to do with where the nucleus is in the cell. We also know chromosome abnormalities are incredibly common in those very first few cell divisions, and the majority will correct themselves. And the reason this has come to light is when they have tried to increase the success rate of IVF, and there's been huge controversy in PGS because you get so many abnormal embryos, which actually may go on to correct themselves. The other thing that we notice is there is a very high loss of embryos between IVF. So many fewer make it to the blastocyst stage than are present in the morula. And therefore there is a big problem at this stage. And we know that there's an increased risk of, as I said, monozygotic twinning with IVF, but we never ever see these twins in IVF embryos. In the Petri dish, we do not see twinning, even though they're not returned until day 5. So if this diet has already taken place by the time twinning starts, and if you've started to polarize what cells are actually going to be what tissues, if you then halve your embryo, it is not surprising that you are going to get different abnormalities in different parts of the embryo because they've already got different gene expression. So this is a paper that has been produced this year, and the Reunion Islands, I don't know if many of you know the Reunion Islands. I have to say I didn't quite know where they are, but we can see that they are very, very isolated. And those are good places to do studies in. And it is also a French protectorate, and it's treated as though it is part of France, like somewhere like Martinique, and therefore they've got good data. Now, the only thing that is bad about this is they're so remote, they have done virtually no genetic studies in these, but they can do this by observation. So they have taken a few steps to look at that because they know they're missing genetic studies. So they've looked at all the live births that have taken place over about a 10-year period. They've looked at the number of congenital abnormalities, and they have looked at 227 twins, monochorionic twins with congenital abnormalities. 152 of these were dichorionic, and they have actually excluded those, and these are their exclusion. So they were left with only 44, but still it's a very well conducted study. It's only just been published this year. And they had 38 twin pairs who were discordant. That was 45 twins. So there were some pairs that both twins were abnormal, but they were discordant. They excluded the ones where they had identical congenital abnormalities. And in 31 pairs, they had only one fetus with an abnormality. So it really does show identical twins are not identical. So what was the overall rate of abnormality? It was about 10.5%. With, if you think about the general abnormality rate in singleton is about 2 to 3%. And there was more discordance than concordance for fetal abnormalities, and 31 twin pairs, only one was affected. As we all know, there is nothing surprising about what they found. with congenital cardiac disease being by far and away the commonest abnormality. And many abnormalities they only had in one twin, and therefore I haven't quoted exactly what they were, but genital abnormalities were quite common, as were things like spina bifida, limb abnormalities, and the syndromes and likely syndromes were the other percentage that they actually looked at. So they found what is already been provided in the literature, but it was quite nice to see this study, which I felt was very good. So why is congenital heart disease so common in monozygotic twins? And I think obviously we know about the blood flow differences in twins, and things like pulmonary stenosis are probably being caused by a differential blood flow potentially in the twin. But it is quite difficult to understand a lot of the time. But I think I'm going to come back to laterality defects. And the heart is probably one of the most asymmetric organs in the body. And I think this is very heavily involved with the twinning process. One of the things they didn't identify in the study in Reunion Island, because it is still very rare, but cloacal abnormalities are much commoner in twins, in monozygotic twins, than in singleton pregnancies. So twins can remain absolutely throughout them. To be much more like very early on. You would have said environment would have changed things, and it does. And there are many, many studies of twins being brought together and what is the genetic contribution for disease. And twins have played a huge part in looking at that. But of course, that isn't of interest to you because you're only interested in the in utero life. So what are monozygotic twins discordant? And this is the big interest to us all. So X inactivation, I'm going to start with X inactivation. We know that men and women are both normal, but actually women have an awful lot more genetic material than men. The Y chromosome contains really virtually no useful genes except to make you male. There are a few at either end which are similar to the ones at either end of the X chromosome. But because women would otherwise have such an excess amount of genes, so it's as though they would be like trisomy for Downs, they'd have a whole extra chromosome. One X chromosome is inactivated, and it is non-randomly inactivated. But this probably occurs before twinning. X inactivation actually starts, and you can imagine, and this is really well documented, that there is much more skewed X inactivation in identical twin girls. Monozygotic twinning is commoner in girls, and the more you go towards conjoined twins, the more there is a female preponderance. So it may be that non-random X inactivation in itself predisposes to monozygotic twinning, or monozygotic twinning reveals a lot of X-linked diseases. So we know that many, many diseases have been described where the monozygotic twins are discordant for a phenotype. Rett syndrome is an X-linked dominant disorder, which is associated with severe mental retardation. And you can see one normal twin and one abnormal twin purely through X inactivation. Leach-Lyon syndrome is an X-linked recessive disorder, which should only present in boys. But if you get complete skewed X inactivation, then you will get a female affected. And this is a horrible disease where children self-mutilate, they bite off their fingers and their tongues, and they have severe learning difficulty and very high uric acids. And we've seen it in, even in something like hemophilia A, we've seen girls that are affected and the boys are unaffected. So the next thing that I want to actually mention is mitochondrial disorders. I alluded to mitochondria earlier on. So all your mitochondria are inherited from your mother, and we have you have one nuclear genome per cell, but you have multiple mitochondria which have their own DNA per cell. And there is approximately 300,000 mitochondria per oocyte, and that is fixed when you are about 25 weeks inside your own mother's tummy, and there is no more mitochondrial production until the fetal genome is switched on. Not all mitochondrial DNA within the cell, in the ooplasm, is actually the same, and therefore you can get heteroplasmy. You can then get unequal sharing of the mitochondria between embryos, and so the mitochondria, and they're absolutely essential for the metabolism of the cell. So there is probably much we can possibly ever think we can study. So if we just look at mitochondria with two different populations of mitochondria, there will then be, when the oocyte is actually performed, we know with mitochondrial diseases, you get differential amount of mitochondria within a cell. They go through this sort of bottleneck. So you can imagine with twins, if you've got two different populations, you would get different numbers of abnormal and normal mitochondria in each of the embryos. So you could get one normal and one abnormal because of differential proliferation, which occurs after the split of the embryo. So I think mitochondrial disorders have not really been studied as far as twinning is concerned. And if you get a polar body twinning, there is very little cytoplasm within the polar body, and therefore you're going to get less energy produced. And if you have few mitochondria, your growth is going to be stunted until you've actually produced more of your own. So you may have differential size just from your mitochondrial production. Now this is unproven, but a very plausible mechanism of differential expression of diseases. Now, getting on to sort of more genetic abnormalities, we talk about genomic imprinting. Genes, there are a number of genes which are only expressed when they originate from one parent, and therefore a maternally imprinted gene is only then expressed from the paternal copy, and then vice versa. So an imprinted gene is normally a switched off gene, and if you're not the imprinted gene, you are actually switched on. So again, if we go back to when diseases, twinning actually occurs, if it is occurring before the imprinting is occurring before day 5, and then there is a separation, you are going to get differential imprinting within the fetuses. Now, the classical one considering this is Beckwith-Wiedemann syndrome. And Beckwith-Wiedemann syndrome has lots and lots of genetic mechanisms associated with it. This is a condition associated with overgrowth, prominence of the fetal tongue, and omphalocele. We are more and more diagnosing Beckwith-Wiedemann antenatally in view of the possibility of omphalocele, but just one warning that you mustn't use a CVS to look for this because of this differential imprint. We are still relying on, at the present time, on amniocentesis, but that's a by the by. Now, 50% of Beckwith-Wiedemann syndrome is due to loss of methylation on the maternal chromosome. So here you are, the maternal chromosome should be methylated at this stage. This is the chromosome that originated from the mother's side. And if you lose this methylation, this is acting as a paternal copy, and you will get Beckwith-Wiedemann syndrome. And as you can see, it is the commonest cause of Beckwith-Wiedemann syndrome. And we know that there is an increased risk of discordant Beckwith-Wiedemann syndrome in monozygotic twins. Now, we don't know if this is because, and there's an increased risk in female fetuses as well, and this may well be due to the X inactivation, or it may be due to the twinning process. But there is no doubt that imprinting disorders are more common in monozygotic twins than in singletons in proportion to their in IVF twins as well. So there's something going on here which we haven't understood, but it seems to be very important. So what about post-zygotic changes? So your embryo, when all these cells are pluripotent cells, you can go on and each mitotic change, you can get a mutation actually occurring. So you can get a few cells affected or none. Now, if you remember, I talked about earlier on the problems with IVF, and we'll come back to that in a minute. But post-zygotic changes, they can either be chromosomal, or you can have single gene mutations. So you can have either of these in this. So if you look at the pre-implantation embryo, we know that those first few cell divisions result in a high number of polyploidy. So looking at the chromosomes in these pre-implantation embryos, you see masses and masses of abnormalities. So it is not surprising that you are going to get mosaicism in these monozygotic twins, depending, and it's going to be different in each of the embryos. Now, we know that if it occurs in one of the very early cells, you will get it in lots. If you have it in the germline, we know that every cell in that offspring would be affected. But this is what we are looking at. We are looking at somatic mutations. So you're looking at post-zygotic mutations, and you can, if it's in one of those very early cells, you will get it in mosaic in one, or you could get it in both twins, but you could get it in very different numbers. But if it occurs later in life, you would only get it in one spot. So we think that mosaicism is an incredibly important cause of differential abnormalities in monozygotic twins. And then I want to look at setting up gene expression. So we know that all our genes are not active in all the cells. So they have to be differential gene expression. Now we have three body planes. We have the dorsoventral and craniocaudal axis, and that gene expression starts, and it's potentially because the nucleus, where it sits in the cell, and I don't know if that's actually right, but when you set up those axes, it will influence the right-left or the laterality axis, which actually probably starts after twinning, but it is highly relevant to how this has actually set up in the first place. And it is probably the major differences in identical twins will be because of this differential setup of the axis in the two different twins. And because the heart is the most, if you like, asymmetrical organ is it may be the most vulnerable to these defects if you are twinning at a slightly later stage. So I think this probably is the majority of the reason that the laterality, the dorsoventral and cradiocornal axis has been set up by the time that twinning actually occurs. So I think this is potentially the cause of laterality. So what about other environmental differences? And I think that there are endless different ways that gene expression actually occurs. And this will be the major cause of changes in postnatal life, continue to absolutely fascinate people. They've even looked in COVID already at the genetic contribution. They've looked at twin studies in every disease that you can possibly imagine, as it is such an important way of looking at nature and nurture. The trouble is identical twins are not identical in the vast majority of cases. So in conclusion, I would just like to summarize why there are differences in monochorionic twins. We talk about non-random X inactivation, or it may be random X inactivation, but when the embryo splits, they will have a skewed X inactivation in the fetus. Mitochondrial inequality, we've both got heteroplasmy, and so there may be more abnormal mitochondria in one fetus. There may be many fewer mitochondria in one fetus. If you've got an unequal splitting of the egg, you can have unequal splitting of the cytoplasm. We've got genomic imprinting, which may increase the risk of twinning. If there is a faulty genomic imprint, and then you will get discordance. Mosaicism is well documented, trisomy 13 in one twin and not the other. We've got gene differences, and then there is also the in utero environmental, all extremely aware. You may have a differential blood supply, and where how much placenta each of the babies may have. So I think there are, having looked at this in some detail, I'm amazed that any monozygotic twins are actually identical. And there is indeed a huge similarity between the twins. And you must presume the earlier that twinning occurs, it must be a more equal twinning event. But I do think we have to relook at the timing of twinning. So I'm not going to I'm going to end my slideshow now. So if anyone has got any questions, I'd be very happy to answer them. I'm going to stop sharing my screen. I hope that was sort of what you wanted. I wasn't quite sure.
Yeah, that's perfect. That's absolutely perfect, because I think it's certainly that the most of the people on the webinar, they're the clinicians, so they're obstetricians or fetal medicine doctors, and therefore this is very useful, because I think
we're not hearing you, Asma.
Can you hear me now?
Is anyone hearing Asma? We're not hearing you, Asma. Yeah, it's very poor, actually. Shall I look at the chat?
Can you hear me?
No.
Okay, can you hear me now?
Yes, that's better.
Okay. So I was saying thank you so much, Tessa. I think that's really useful. Often people would put their questions in the chat box, but often it takes a few minutes, maybe 5 to 10 minutes, for people to actually write their questions. I think this is very useful. and certainly for the audience, because most people will find genetics very complicated and challenging. Would it be okay, Tessa, if you stay until Laura does present her case, and then maybe we can have the discussion together?
No problem, no problem.
Okay, great, thank you so much. So Laura, are you okay to share your screen?
You're on mute, Laura.
I'm gonna unmute her.
I'm unmuted. I'm unmuted. So yes, that's okay. Can you see my screen?
Not yet.
Ah. And now, can you see it?
Yes, we can hear. We can see yours.
Great. Okay, so good morning, everyone. Today, Asma asked me to present you a clinical case so about twin pregnancies, and we have decided to present you this this case of a twin reverse arterial perfusion or TRAP sequence. And it is women who was referred to St. George's Hospital for a second opinion, and she is gravida 2 para 0. She is 40 years old. It is a spontaneous pregnancy. She has a high BMI of about 34. She is rhesus positive, and on the first trimester screening, there was a diagnosis of monochorionic diamniotic twins with one of the twins, and the other twin had a normal nuchal translucency that was she is referred at St. George's after the anomaly scan for a suspicion of TRAP sequence, and she arrived at George's at 20 weeks and 6 days. So the first twin had a normal heart activity, but this twin was really small. So you can see here all the biometries are well below the 5th centile, and this twin had a weight estimated at about 190 grams at 20 weeks. And it also presented with oligohydramnios with a deepest vertical pool of 2.2 centimeters. And you can see it on this picture. So this is the first twin with the membrane between the endocrine membrane. And you can see that there is, right, you can see the second twin. That second twin did not show any heart structure and heart activity. So it was grossly abnormal, and we estimated the fetal weight of this twin at about 250 grams. And that twin had a normal amniotic fluid volume with a pool of about 5 centimeters. And I'm going to show you a short video of this twin. So we could confirm that it was a cardiac twin. You see here this malformed second twin, so there is no heart. There is here a feeding blood vessel, and you can see that there is a kind of cephalic pole that is appearing on the screen just now, and there are some kind of inferior legs also, and this main feeding vessel there. So after this first scan, we could confirm that it was a monochorionic diamniotic twin pregnancy that was complicated by a twin reversed arterial perfusion sequence. The first twin is the pump twin, the second twin is the cardiac. But on top of this TRAP abnormality, the pump twin here was affected by growth restrictions. So his biometries were far below the 5th centile and cardiomegaly. And on this picture, you can see the Dopplers of the pump twin. So to the left, you have the Doppler in the umbilical cord, and you can see that there is absent end diastolic flow. To the right of the screen, you have the Doppler of the ductus venosus, and you can see that the A wave is negative. Then our cardiologist accepted to see the patient on the same day, and they confirmed that there was cardiomegaly, as you can see on this short video, with a mildly hypertrophied right ventricle, but the heart of this pump twin was structurally normal. On this picture, you can see the Doppler waveform of the feeding vessel in the cardiac twin. And this is the Doppler waveform in the umbilical cord of the cardiac twin. And you could see that for this twin, there was a positive end diastolic flow in the umbilical artery. So after this first scan, we had a long discussion with the patient, and we explained her that the prognosis was extremely guarded for 2 reasons. So the first one that the pump twin can you hear me now?
Yes. Yes, we can.
Okay, great. So yes, so I'm going to start again maybe. So after this first scan, we had a discussion with the patient, and we explained her that the prognosis seemed extremely guarded because there was this TRAP issue, but the pump twin was also affected by early onset fetal growth restriction. And the cardiac twin was above 50% of the mass of the pump twin, which is one of the usually one of the most commonly used prognostic factors for TRAP. And so we discussed 3 management options. The first one was expected management. The second one was the termination of the pregnancy due to this really poor prognosis. And the third option was to try to intervene and to do a laser photocoagulation of the blood flow in the cardiac twin. The mother was really committed to the pregnancy, and she opted for the intervention. And so we performed the interstitial laser on the cardiac twin on the same day. This was quite advanced gestational age because she was already 20 weeks and 6 days. So we knew that there was a higher rate of failure for the procedure, and we asked her to stay overnight to check that the procedure was successful on the day after. But unfortunately, on the next day at the post-laser scan, there was a persistent blood flow in the cardiac twin, as you can see here on the picture, and the pump twin still had abnormal Dopplers. So and at this time there was a reverse flow in the umbilical artery, but on that day there was a positive A wave in the ductus venosus. So once again, we discussed with the patient, and she was absolutely committed to the pregnancy, and she wanted everything to be done for this fetus. So we still offered expectant management, but she was really keen to have a repeat laser photocoagulation in the cardiac twin. So we performed this repeat laser, and at the same time, we took some amniotic fluid, and we confirmed a few hours after the procedure that this time the blood flow was well interrupted in the cardiac twin. But then on the follow-up, unfortunately, there was a demise of the pump twin 5 days after the procedure. And when the results came back, the amniocentesis showed that this pump twin was affected by trisomy 13. And so that's why we wanted to discuss this clinical case, because when this was an unusual presentation for a TRAP sequence, because the pump twin is not commonly growth restricted. And that reminds us that, every time we see an abnormality, there can still be, even if we have a TRAP sequence, there can still be an aneuploidy, an underlying aneuploidy. And so to open the discussion, I've searched the literature to see if there were a description of an association between TRAP and aneuploidies. And I've only found a few publications. This first one that is quite old, but that showed studies. It was a review of studies where they had looked at chromosomes in both the pump and the cardiac twin. But as you can see here, most of the time the abnormal set of chromosomes was found in the cardiac twin, and most of the co-twins had a normal set of chromosomes. Apart from a few cases where they showed a sex aneuploidy, and it was in this review every time a Klinefelter syndrome. Then I found a much more recent publication in 2000 about this association of TRAP and Klinefelter syndrome that was found in both the pump and the cardiac twin. But the same authors also had a second case where this Klinefelter syndrome was only found in the pump twin with a normal cardiac twin. Then I've tried to find associations between TRAP and trisomies, and I've only found 2 papers that were describing an association of the TRAP with trisomy for the second chromosome, but each time this trisomy was affecting only the cardiac twin. And one was in a triplet IVF pregnancy, and the other one in a spontaneous twin pregnancy. And so the authors of these 2 publications discuss a possible explanation for this association of TRAP and discordant aneuploidy. The first 2, Tessa has already described them, and much better than me. They said they discussed the possibility of a post-zygotic non-disjunction that would explain that the triploidy was only found in the cardiac. They also discussed the possibility of a fertilization of the ovum and of the first polar body. But they also had a third mechanism that was discussed, and that was the possibility of a fusion of the 2 placentas really early in the first trimester, with then the development of artery-to-artery anastomosis that would then lead to the TRAP sequence. And so in that case, the twins could be, of course, non-identical for their set of chromosomes. So yeah, that was the presentation of this case of TRAP and trisomy 13. And thank you very much.
Okay, thank you so much, Laura. Can you hear me, guys? You can hear me?
Yes.
Fantastic. So Tessa,
yes, I can hear you.
Tessa, let's start with what's your comment on this case?
I think it's certainly, I think with TRAP twins, from my understanding, is that they can tolerate chromosome abnormalities that would otherwise be lethal, like the trisomy 2, because they're being supported by the other twin. So from the point of view twin, why it is actually survived, which it wouldn't survive otherwise if you call it a survival. I don't think the trisomy 13 is probably the cause of the monozygotic twinning here. I think it is probably incidental to this, because we are not seeing increased monozygotic twinning in chromosomally abnormal fetuses. So I think it's probably incidental, but I suspect the trap is just only surviving because of you didn't do the chromosomes in the trap twin.
No, so unfortunately we don't know if the trap twin was also affected.
But Laura, I think that actually the amniotic fluid was taken from the trap, because it's a diamniotic twin pregnancy, and when we do the laser, we go inside the sac of the trap, not the sac of the other one.
Yes, but that's yeah. I don't. That's what I know, but I thought, well, that in what we described, we had taken fluid from the pump after the laser, like in a second.
I mean, I suspect it would be from the amniotic sac of the trap, because you will not go with a laser into the sac of the pump twin. You'll go for the trap. But actually, even though the pump twin was very, very growth restricted, and therefore, I suspect this baby also had very poor prognosis. But I suspect the trisomy 13 was in the trap.
I suspect it was in both, actually. I suspect, because that's why the pump twin was so abnormal, don't you? I suspect that was the cause. And we know that actually the majority of trisomy 13s will miscarry, won't they? We know the first thing, I'm surprised it got as far as it did, is the only thing, isn't it?
Yes, so and this case I think raises a number of points, because she had first trimester scan in her local hospital, which I suspect they probably saw no cardiac activity in the trap mass, and therefore they thought maybe it was a twin and single intrauterine demise early in pregnancy. And the message is that whenever you have a twin pregnancy in the first trimester, and when you have a single intrauterine demise, you think that actually one twin died early, you need to put the Doppler on to make sure that it's not a trap, because when you put the Doppler, you will see the reverse flow in the trap mass. We don't miss it, because I think she was only diagnosed at 20-week scan when she came back for the 20-week, and then you find that actually the bump twin, or one twin, is very small, and then there's another twin which was about 50% of the size of the other one, but also had the reverse flow. Okay, so Tessa, can I just ask you, because it's I think a lot of people find it very complicated or difficult to understand the mechanism of the heterokaryotype monozygotic twin pregnancy.
Sorry, what did you say? Something monozygotic. I missed one word there.
Heterokaryotype monozygotic twin pregnancy.
So where you get different karyotypes within the two twins.
Yes.
And I think there are two possible mechanisms for this. The first is that you get something called trisomic rescue. So you can start off as a trisomic fetus. But what you have to remember is that chromosome abnormalities, if you remember from that slide that I showed of IVF, pregnancies undergoing pre-implantation genetic screening, have the most phenomenal amount of chromosome abnormalities. And these can go either way. So if you start off with an abnormality, you could go back to normality. So every time you have a cell division, you could potentially land up with an extra chromosome or a missing chromosome. And remember, normal cells tend to replicate better. They have better replication. So you could get what we call trisomic rescue. So you get one twin that goes back to being normal. Is the abnormalities occurring mitotically. So that's in that ball of cells with that one where I showed that one cell has got a mutation that has occurred. Remember, it is chaotic, those first few cell divisions. The number of abnormalities is enormous. And I think that if you twin with that abnormal selection. So I think it is most likely that the vast majority of the abnormalities are occurring after conception, when you've still got the ball of cells, the blastocyst or the morula, that actually just mutates at that stage. So I think that actually, if you then think that twinning occurs after that, you can actually see how you will get abnormalities in one and not the other. If you're speaking, we can't hear you.
Okay, so can you hear me now?
Yep.
Perfect. So, and therefore, for the simple clinicians and in among the delegates here, if you have a monochorionic twin pregnancy with discordant anomalies, so one baby is abnormal and the other one is normal, do you recommend to do double CVS if it was in the first trimester? Well, one CVS because you have one placenta, double amniocentesis, or just amniocentesis of the affected twin?
Well, I think it slightly depends on what the abnormality is. There are certain abnormalities that actually have a low chromosomal association. So if you had a spina bifida, I mean, we have seen it in triploidy, of course we've seen it in trisomy 18, you may take the view that it is safer to just do one, but fundamentally you should do the karyotypes on both of them. You know, I think that you should. And this is you know, when you're doing laser for twin-twin transfusion, you are not expecting an abnormality. So I think it is perfectly reasonable under those circumstances just to do the QFPCR on one twin, because actually we're not really looking for a chromosome abnormality. But if you have got an abnormality in a twin, I think it is unsafe not to do a karyotype in both. I think the difficulty arises in the diamniotic, dichorionic twins. If they are of the same sex, I think there is actually, I think the monochorionic ones in many ways are quite easy. I think it's the other ones that are much more difficult. Certainly we had one recently, didn't we? But it was a monochorionic and they both had Down syndrome. So I think that actually same-sex twins should probably both have investigations. Now, of course, at 12 weeks, you don't know if they're same-sex twins, do you? And so you could take the view that you would only do the second investigation if the first has been abnormal at 16 weeks, when you can look at the you know, the karyotype. So I think that it does mean that you're going to have a sequential testing, but I would be influenced by you of the increased risk of miscarriage over a single via double procedure.
Okay.
And I think that's where it really goes down to.
Yes, and in fact, actually, Tessa, you mentioned about this case that we had recently, and we had discussions. This was a case of dichorionic triamniotic triplet pregnancy, where they actually put, during the IVF, 2 embryos. So we know that one embryo split, and one of them was had increased nuchal translucency and had abnormalities in the first trimester. We did the CVS and selective termination of this one, and that came back as Down syndrome. And later, at 16 weeks, when we discovered that the gender, there's one, the monochorionic one, sort of, or the identical twin of this, of the one that had the trisomy 21, and we tested that, and it came back as trisomy 21, despite the fact that the scan was normal. There was no abnormalities on this baby. And therefore, I was really surprised about the fact that you could have a twin pregnancy. They both have trisomy 21, but they actually have different phenotype. So one of them have all the characteristics, big nuchal, heart abnormality, all the markers, and the other one looks completely normal on ultrasound scan, but also have trisomy 21. How do you explain it genetically?
Well, I think that actually you've got all the other mechanisms of the genetics of that splitting of that embryo. And I suspect mitochondrial abnormalities or mitochondrial differences are probably very important here. So if you've got one embryo that refuses, who receives very few mitochondria, you can imagine that is a much more compromised embryo. And if it is on top of it has a trisomy as well, it may well be metabolically much more likely to develop all the abnormalities. So I think that you are unlikely with trisomy 21 to get things like imprinting or any of those other mechanisms. But I think the mitochondrial issue is probably much more relevant. So that's, and it's totally unproven, but I think that you can understand how that could be hugely relevant.
Yeah, but that's, I mean, should we change our practice? Because so far, if we have a dichorionic twin pregnancy, one of them have big nuchal, an abnormality or increased risk, another one looks normal, certainly a lot of parents or a lot of even clinicians would probably suggest to test, do CPS for the affected one and don't test the other one really. So what are you saying is that if this woman come back at 16 weeks and the other twin have the same gender, same sex, should we also be testing the other one? Because it could be monozygotic dichorionic twin pregnancy.
Yeah, I think this is really difficult, isn't it? Because actually the history doesn't suggest to us there are many abnormalities. So we're not getting postnatally. So theoretically, it's absolutely true, but actually the evidence doesn't really support it. And it may well be that if you are a monozygotic or a twin pair and you've both got chromosome abnormalities, you're much more likely to miscarry. So it may be that there is a much higher miscarriage rate, so we are not being exposed to that abnormality. So I think that if you theoretically you should be, but practically I'm not reading lots of cases where it's actually been missed. And so I think that we've got to take evidence that, you know, that actually we shouldn't be doing it. So I think that if they are same sex, you need to always consider that it might be an issue, but I don't think you should be potentially sticking needles into people unnecessarily.
And that's very clear. And what about if it's monochorionic diameter twin pregnancy with increased nuchal in the first trimester, but actually there's no abnormalities? And sometimes we say, well, maybe it's actually a sign of twin-to-twin transfusion syndrome. Suppose it's not TTS. Do you think we should be testing this pregnancy? And if we test, should we do double amniocentesis at 16 weeks?
I don't know. I think it's there's so many of the monochorionic twins do have raised nuchal with one of them, don't they?
Yeah.
That again, because we're not seeing chromosome abnormalities in a large group of these, I think that what you're doing appears to be correct. And as I said, if they are more likely to miscarry because they are twins with a chromosome abnormality, I mean, let's face that one you have just presented, that was never going to live, was it? It was actually not going to survive that pregnancy. So I think that stick more needles into twin pregnancies. Actually, bearing in mind you may well end up having to do laser later on. Personally, I don't think you should be putting extra needles into these people. I think what you're doing seems to work, and I don't think you should change your practice on theoretical risks because I think that's dangerous.
Okay, great, thank you. Very clear message. There are some questions. Tessa. So from Rohan, is there any role in the expression of surface protein genes in post-zygotic cells, which could drive the development of monozygotic twins?
Oh, I've got no idea. I'm afraid surface protein genes, surface protein of the oocyte, or what's I'm not sure what you mean by surface protein genes. I'm afraid, because there are we have not looked at any. We have got no genetic mechanism for inherited monozygotic twinning. And I don't know why they haven't looked at this armadillo a little bit more carefully, because it must be actually just staring us in the face what it actually is. And I think unless you actually could look at the ones that are inherited, I don't think we're going to find out further about that, because you're much more likely to be looking at the expression of these genes rather than actually mutations within them. Personally, I don't think that we would actually get that. So I don't think you'd get mutations, but I think you could. So I think we need to all go out into the bush and collect our armadillos, which will help us with that.
It's great. And just as a follow-up with this question, Tessa, so you said that twinning never really occurs in a Petri dish. So when you're doing IVF and you, let's say you transfer day 5, you never have twinning or splitting, and it only happens inside the uterus or inside the human body. Do we know why? Or do we have any potential explanation or any theory even?
No, and it suggests that twinning happens later. It may be that actually twinning just does not happen at that very early stage. And if we have got the whole twinning wrong, we've got the whole cause of the twinning wrong. And that's why I think it just needs to go back and start again, looking at when twinning occurs. And I think the real problem is we have very few models of twinning to actually look at that, because, even with sort of the armadillo, it's quite difficult to go on killing armadillos, isn't it? And there are not any good animal models, and they don't seem to be able to get animals to twin. And you don't have Drosophila, I don't think, twins. So I think the studies have been difficult to look at it, but I think the whole supposition of when twinning occurs is probably wrong.
It's interesting because a lot of the talks, and you showed this slide, the typical slide of actually, the type of twin pregnancy and according to the time of the splitting, which I think we take it for granted, but actually you would say that this is probably questionable.
Yeah, and the chap who quoted the paper, he just said, once we've got IVF, this can be rewritten. But nobody, and when I was doing that twin chapter a couple of years ago, there was only one paper that had actually questioned whether it was actually correct. So I think it is always quoting the same papers and then going back to the original paper. And I would say that to everyone, of going back to original papers is always quite instructive. And I remember with agenesis of the corpus callosum, of the incidence in the general population, and this was all identified on air encephalograms. I mean, an air encephalogram was the most terrible investigation. Most of you are too young to have even heard of air encephalograms, but actually the detail of the brain was absolutely minimal. So I think there's a lot of dictate out there that actually isn't true.
So we need more research, more studies in twins. Vetrana Carrick, I don't know that you remember Vetrana or not, Tessa, but Vetrana used to be a registrar at St. George's Hospital, and now she's consultant in obstetrics and fetal medicine up north. And in fact, she obviously sort of sent us some complicated twin pregnancies, including TTTS. So she had a question. She said, I had a case of MCDA, twin pregnancy, increased nuchal, and had a laser fetocide Turner syndrome. The other twin boy was Turner mosaic. Should we offer cord bloods for cytogenetics in MCDA with one twin having increased nuchal?
I don't think so. I think that that is very well described of 45XY, 45X twins because of the discordant sex, which of course was the most obvious. And it is, you know, people love to find these crazy cases. But I don't think there is necessary. I mean, obviously, if you've got a Turner's and you've got you know, a high nuchal in a Turner's or something like that, but I think I would want to confirm the abnormality in one twin. I certainly wouldn't think that cord blood was necessary. And of course, cord blood, incredibly importantly, is unreliable in monozygotic twins because of mosaicism in the blood and mixing of the blood. So actually, I would say categorically no, because you can absolutely get the wrong results because of mixed, you get chimerism. And so actually you've got to be very careful about blood studies in monochorionic twins because of the mixing of the bloods.
Okay, so very clear message there. So I think the one challenge that sometimes you face is that if you have a monochorionic diamide twin pregnancy with one of them is hydropic, lots of abnormalities, and let's say the parents decided to have selective fetal-side termination on this one, the question is that should we test the other one? Should we actually stick a needle in that normal, because we try to avoid the sac of the normal twin. And to some extent, the one that hydropic has poor prognosis, and therefore you're going to just do selective fetal-side on this one. Should we actually be testing, let's say, taking amniotic fluid sample from the normal twin?
Well, I think that you can look at that two ways. You're doing the testing on the abnormal one. You would get an array on the abnormal one because the fetus is hydropic. It isn't just got a raised nuchal translucency. If there is an abnormality on that one, then you should go back and check the other one. But it depends whether it is safer to do the procedure at the same time, which might be completely unnecessary, or you do it sequentially only if it is necessary. And I would have thought it was better to do it sequentially. Now they may refuse, but you know, like the one with the trisomy 21, she was very keen to know. And so I think I would wait and do them sequentially, but I would have to look at the miscarriage rate or trust you with what you feel is the safest thing to do. And I would have thought it was safer to go in again at the second time. I'm always amazed when we've had to repeat invasive tests. So we've started with a CVS, we've got mosaicism, and then we've done a second test. I can't remember a miscarriage on those women that have had a second test. And that always, is it because you would miscarry on the first test? You know, and therefore it seems remarkably safe now. You know, I don't know if that's the case.
Yeah, certainly I agree with you. I mean, I think our, you know, relatively obviously limited experience with cases where you need to do repeat invasive testing in twin pregnancies, I don't think this necessarily means that a lot of them miscarry or rupture the membranes, especially in this scenario, as you mentioned. So last question, what do you think, what's your view, your own view, and maybe comment on the UK practice of PGT or pre-implantation genetic testing for an employee in IVF pregnancy, particularly if they're going to put two embryos back?
I think it's very interesting, PGS, and some people, if you talk to our IVF colleagues, they're quite pro PGS. And I think it's better than it was in so much as you only used to biopsy 1 or 2 cells. And therefore, if you've only got 1 cell, it's really not telling you a lot about that embryo. Now that they biopsy them at day 5, you've got a few more cells that you are actually looking at. But the number of abnormal cells within those cells, and so there are now a lot of units which will reimplant an abnormal cell if it has an improbable karyotype. So I remember someone saying to me, well, shall we reimplant this with trisomy? I can't remember what it was, trisomy 5 and monosomy 5. You know, there was one cell with one and one cell with the other. And it was felt that actually the rest of the embryo was likely to be normal. But if you are going to reimplant an abnormal embryo, why have you done it in the first place? So I think I feel that it would be probably better to do very selective testing of the embryo. So if you selected it for 13, 18, 21 and the X chromosomes, X and Y, you would probably do most of the good that you possibly can. And so I think the problem is with full screening, and they're now looking at screening, you know, for array CGH on these. You're not going to really have enough embryos to reimplant. And bearing in mind they're often older women, I think this is tricky. So I think the jury remains out of whether it is of any value at all.
And certainly the practice varies. So I think outside the UK, I think I see many women who had IVF and they would have had at least BGS, for example, if not the whole testing. In the UK, it's rare actually to see them having had testing.
Yeah, and I think it's the number of embryos that are actually produced, and they will only do it if you produce a lot of embryos. So if you produce a lot of embryos, you can say, well, I can afford to waste quite a lot of embryos. But if you only produce 1 or 2 embryos, I think in most places they would consider it not appropriate. So I think it's difficult, and I think we need to wait and see more studies, because PGS is, you know, it's an expensive procedure.
Yeah, and the final question: if the woman had a testing before, would you still do NIPT?
No. I think that when you look at doing NIPT and PGS, I think that you should actually trust one or the other. So I think that, you know, I did have one woman who'd had PGS, she had had PGD, screening for recessive conditions, and then she did NIPT, and it was an XY mosaic. It was there was a sex abnormality. And so we did do an amnio, and we did find that there was an XY-XO mosaicism. And so that is going to be post-zygotic. Actually, I persuaded her to keep the baby. I said, look, this is a super precious. This was the only embryo that was actually viable. And so I think that actually, and the baby was completely normal. It was a male, and it seemed absolutely fine. And so it was the right decision. So I think that NIPT was actually unhelpful to this woman. I think actually she'd been much better off of not having NIPT. And so I think if you can over investigate these women, and I think that I would say that you have to stop and not do every test you possibly can do.
On this note, I think this is a very good message to finish this discussion. Thank you so much, Tessa. That was really, really useful for such a, I think, a very complex and I think difficult. Most clinicians find it really difficult when you have this really confusing results or sort of genetic results, or how do you handle complex twin pregnancies. And thank you so much, Laura, for Noel, who is a consultant in Belgium, and she's doing some fellowship with us at St. George's. Really nice and short, you know, at short notice, she did this interesting case with quite unusual presentation and surprising results. So thank you so much. Thank you both, and thank you for all the rest of the people on the webinar. The next webinar in 2 weeks' time, and we will have also interesting cases by Surabhi Nanda. And also one presentation by the key speaker, and then followed by cases and discussion. That's the usual style that we have for these webinars. On this note, I would like to thank everybody. Enjoy the rest of the weekend, and thanks so much for joining.